We're going to take a look at endo and exocytosis in this lecture. In vesicular transport, we're going to be using a vesicle, a membrane-bound sac, to move things into and out of the cells. So we're going to use this to move larger molecules like proteins, and this is an active process, so we do need energy to do endo and exocytosis. Remember, endocytosis is when we're moving things into the cells, and there's a couple different types. And exocytosis is when we're moving things out of the cell. We'll start with endocytosis. And one of the types of endocytosis is called phagocytosis. This is done by some of the white blood cells in our body. Phago means to eat, and cyte means cell. So it literally means cell eating. So the white blood cell is going to find some sort of solid particle, maybe it's a bacterial cell or it could be dirt in your wound or something like that, and it's going to surround it with its membrane and then take that inside the cell and pinch its membrane off. Okay, so now you've got this vesicle, this little membrane-bound sac surrounding whatever it's taken inside. And then the lysosome, one of our organelles, will fuse with the phagosome, and the digestive enzymes in the lysosome will then break down and digest whatever was ingested by the white blood cell. Phagocytosis is not specific, so the white blood cell is going to eat anything foreign. It doesn't care if it's E. coli bacteria or salmonella bacteria. It's going to eat whatever it finds, so it's not very specific. And this is just showing you an animation showing the membrane surrounding the solid particle, maybe a bacterial cell, and then we've got the lysosome fusing and releasing the digestive enzymes and then digesting whatever was taken in. Pinocytosis is cell drinking. Pino means to drink, and all of our body cells will do this. So they're going to just take in some of the extracellular fluid and whatever's dissolved out there, so whatever solutes, and they'll just take that in and they're just sampling their environment to see what's out there. And so all of our cells will do this. And again, it's non-specific. So just whatever is in the extracellular fluid, that's what gets taken in. And the last type of endocytosis is very different from phagocytosis and pinocytosis. This is called receptor-mediated endocytosis. So this one's different because it's very specific in what gets taken into the cell. So we have some specific receptors that are out on the cell membrane. And for example, we take in the LDL, the bad cholesterol, into our body cells by receptor-mediated endocytosis. So these receptors will just bind to the LDL, the bad cholesterol, and when it binds, that will stimulate some proteins on the inside of the membrane to start folding, and that will bring the membrane in and pinch off. And that's how you bring in the LDL, those low-density lipoproteins, into your body cells. So this one's very different because it's very specific what is brought into the cells, whereas phagocytosis and pinocytosis are not specific. Exocytosis is the opposite of endocytosis. So we're going to be moving these larger molecules to the outside of the cell. So for example, maybe this is a pancreas cell, and we've got the protein hormone insulin, which is stored in these secretory vesicles, these little membrane-bound sacs. And then when the pancreas gets the signal to release the insulin, the vesicle will fuse with the cell membrane, become part of that, and then the insulin will be dumped out into the extracellular fluid. So we're going to see exocytosis also being used to release neurotransmitters in neurons. And we also release antibodies from white blood cells this way too.